A very good morning to everyone. My name is Priya. I am the Assistant Professor of IIMT College of Pharmacy. In today's lecture, we're going to discuss about spoilage. In this lecture, we're going to discuss about these are my content, which is we are discussing in today's lecture. What is spoilage? Type of spoilage, factors affecting the microbial spoilage of pharmaceutical product, and lastly, the sources and types of microbial contaminants. Firstly, when we talk about spoilage, we have to understand what is spoilage. Spoilage is the chemical and physical degradation of pharmaceutical product which tends to or which rendering it unsuitable for use. If we define spoilage of pharmaceutical products and drugs are referred as the changes in the physical chemical properties in such a way that the formulation or therapeutic agent get deteriorated and become not suitable for use. Secondly, if we talk about microbial spoilage, it includes the contamination of pharmaceutical product with the microbes, which leads the spoilage of product that affect drug safety as well as the quality of drug and not intended for the use of any consumer. Shortly, if we define or shortly, if we understand what is a spoilage, it is the deterioration of any product by contaminants of microbes. Next slide, the topic is why to study spoilage. Firstly, when we talk about spoilage, that means we have to understand it is the physical and chemical degradation of any particular product. That is why first point is to determine the microbial contaminants present at that particular formulation. Second, to avoid the health hazards of infection, because when you talk about the health hazard, that means it directly indicates the consumers. If the consumers take that spoiled product, it may lead serious health hazards like toxicity, infection, even death also. Thirdly, to acquire the knowledge about the sources and different types of microbial contaminants related to different doses and deterioration of action. uh, like it is not suitable for pharmaceutical industry because deterioration of drug or excipient occurs. As a result, the product may loses its quality as well as the quality, and it may be ineffective. Right. The entire sometimes in pharmaceutical industry, the entire batches may be required to discard, and it may directly cause the entire reputation of that particular company, may damage the product. The damage of that particular product due to spoilage, it indirectly damaged the reputation of that particular industry or particular pharmaceutical company, because there is some there is financial loss as well. And if the spoiled product, or if the company has batch to batch spoiled product, or batch to batch spoiled product are released by that particular company, there is a direct effect in financial loss also be noted. These are the types of spoilage. Firstly, in this flowchart, we're going to share about we're going to discuss each and every types of spoilage. First is chemical spoilage, second physico-chemical spoilage, and biological spoilage. These are the major types of spoilage in case of pharmaceutical industry. First, chemical spoilage. When we talk about chemical spoilage, we have to understand that the chemical spoilage means the deterioration of chemical nature of the drug and excipient. If the deterioration of drug and excipient or molecular if the deterioration of chemical nature of drug and excipient occurs, it may directly lead to the molecular structure of that, to the change of molecular structure of that particular ingredient, that may be drug or that may be excipient. This may also affect the physicochemical properties of that particular preparation. That means this change of the change of molecular structure also affect the physicochemical properties preparation and as well as the potency of that particular drug. The potency of particular drug may be decreased or may be enhanced by this chemical spoilage. The physicochemical spoilage, when you talk about physicochemical spoilage, it may change by the or it may means the physical and chemical properties of that particular product has been changed. The following types of changes may occur. First, viable growth. When we talk about viable growth, it may occurs the inside in the container. The they may be visible in the forms of floating layers, turbidity, etc. The contamination of product by fungus species like Aspergillus species may cause this kind of spoilage. Second, the gas production. Some microbes may produce gas inside the container. These gases may be visible in the forms of bubbles, floccules, etc. The contamination of product with bacteria like E. coli may produce gas if it contains sugar. The coloration and discoloration or decoloration of the coloration and decoloration. Some microbes may decolorize the formulation or it may produce unique color which is different from the normal color of that particular product. The odor formation, the microbial growth of finished product may produce bad odor or characteristic odor, or it may produce the characteristic rotten smell due to physicochemical spoilage. Lastly, the taste change. If you talk about taste change, in mostly in oral formulation, it may impart bitter or obnoxious taste to the oral formulation. Biological spoilage. Third type of spoilage is biological spoilage. It means the spoilage of pharmaceutical product may produce some undesirable and dangerous molecule which has undesirable biological effect. This microbes or this kind of biological spoilage some microbes may produce toxins, pyrogen, or other harmful metabolites as well. Factors affecting the microbial spoilage of pharmaceutical product. There are so many factors which affect the microbial spoilage. Some of these factors reduce the rate of spoilage, whereas some factors increase the rate of spoilage. The factors are related to nutritional requirement of microorganism, environment, and nature of that particular microorganism. These factors may be studied to minimize the impact of spoilage, and some factors are mentioned here. The following factors, the number one factors is the number of contaminating microorganisms. First, they may not be sufficient. There may not be sufficient spoilage if the number of contaminants are less. This slow the label, it contaminants may not be able to multiply due to design of this formulation. However, during long storage of these microbes may cause the damage. If the initial contamination is high, then it may present in undesirable challenges. The products will be damaged quickly due to presence of high number of microbes. High level of contaminants occurs due to lapse of manufacturing process or due to high loads of contaminants in the raw material as well. Second, the types of microorganism. Some types of microorganisms are more aggressive than others. They can quickly multiply and spoilage is much faster. However, aggressive microbes may not multiply initially and due to presence of some preservatives and other substances. The contamination of product with the natural communities of non-aggressive microorganisms can facilitate the growth of other aggressive contaminants as well. Third reason is temperature. Storage of temperature or a temperature storage as is the great controller of microbial growth and spoilage. However, the spoilage over the range like minus 22 degrees centigrade to 60 degrees centigrade, the microbial spoilage or microbial growth is low in a temperature, in low temperature, and as well as in high temperature. The if we monitor the product which is stored in cool place like 8 degrees centigrade to 12 degrees centigrade may cause negligible spoilage. If in case of high temperature can also prevent the spoilage. For example, water for injection is stored above the 80 degree before filling and sealing, so the extreme temperature minimize the spoilage rate as well. Third, pH. The microbes prefer neutral pH for their growth. However, some microbes may prefer slightly acidic pH, and extremely acidic pH or alkaline pH of formulation may prevent the microbial growth and subsequent spoilage. Some communities of microbes can survive extreme pH as well, and the change in pH. They support the growth of other microorganisms as well. Lastly, fifth point is containers and packaging. Enhance the contamination of single dose ampoules and vials are negligible. However, the multi-dose or multi containers may be minimize contamination. If, however, if we talk about multi-dose containers, be contaminated by using themselves, changing in design of these containers minimize the contamination rate as well as the spoilage rate as well. For example, wide mouth containers for cream and ointments are replaced with the narrow tubes with screw up the cap closers. Multi-dose injections are stored containers which have self-sealing rubber wrap. Containers are sealed in such a way that containers are sealed in such a way that it can prevent entirely of water and oxygen to minimize the activity or redox potential. In next topic, next slide, the sources of contamination and the types of contamination. The microbes are a very important part of our environment. These present or they present almost anywhere and everywhere. So the contamination of pharmaceutical product, contamination may occur in pharmaceutical product in a large scale, in, large scale in manufacturing, in packaging, in sealing, and etc. In transporting as well. So, in a small scale of hospital manufacturing during his use of patient, following there are different sources of microbial contaminants in case of pharmaceutical industry. In large scale of manufacturing, as well as medium and small scale of manufacturing, contamination may occur from the following sources. First is water. Water is the major source of contamination. Common waterborne microorganisms like Pseudomonas, acrum bacteria, and other low-demand gram-negative groups may present in potable water as well as purified water. Ion exchange column may be contaminated by water source, and microorganism may multiply the contaminants of purified water. Water which purified by reverse osmosis process, they also may be contaminated, because if the osmosis membrane is not properly disinfected. Even distilled water may also be contaminated if it is stored by long day and in a normal temperature. Second, raw materials. The pharmaceutical products are prepared by varieties of raw materials. Some earth material like bentonite, kaolin, etc. may contain anaerobia spores like Clostridium species. For example, starch may contain coliform of bacteria like E. coli, and animal products may contain variety of bacteria like E. coli and Salmonella typhi as well. The air of manufacturing area. Air is filled with the billions of suspended particles and microbes. Fungus spores like Penicillium, Aspergillus, etc. Bacterial spores like bacterial Bacillus species, etc. also prevent. These spores and microorganism may be contaminated per a pharmaceutical product. This type of contamination minimized by practice of manufacturing and cleaning room in aseptic area under continuous flow of sterility or sterile air through HEPA filter. Personal some manufacturing the contamination may be caused by the in case of pharmaceutical industry, the contamination also caused by manufacturing staff. The person may be infected by various type of infection like bacteria, like fungus, like virus as well. For example, bacteria like Staphylococcus, Staphylococcus, and Actinobacteria, Candida bacteria, they may affect or they may infect any person in the pharmaceutical industry. This contamination may be minimized by the proper health checkup, vaccination, hygiene of personnel, and protective gear, and proper training with the personnel may be minimize the contamination rate and uh, with the help of proper gear and proper training, a person may also minimize the contamination due to which is occurred by pharmaceutical person. Lastly, thank you for listening in this video, and I wanted to say keep calm and enjoy the microbiology. Thank you.